Hello, Dr. Sturms. This is what I ate in one day to deadlift 1,104 pounds. First, I had breakfast. I ate yogurt and strawberries with green tea with lemon and honey. This came out to 276 calories and included 42 grams of carbs, 6 grams of fats, and 14 grams of protein, as well as 20 milligrams of cholesterol. For lunch, I had Hubble. I ate a chicken fried steak, which I learned is not chicken, mashed potatoes with gravy, and roasted broccoli. This totaled 852 calories and 86 grams of carbs, 47 grams of protein, 42 grams of fat, and 138 milligrams of cholesterol. For a snack before 2 hours of organic chemistry tutoring where nobody came, I had everything bagel crackers and some cheese. This was 275 calories, 19 grams of carbs, 12 grams of protein, 11 grams of fat, and 40 milligrams of cholesterol. I kind of forgot about dinner until it was late, so I went back to Hubble and got a cheese quesadilla, cucumber, and then I ate 3 Oreos, like actually 3, not 7. My dinner totaled 897 calories, 77 grams of carbs, 33 grams of protein, 67 grams of fat, and 101 milligrams of cholesterol. In total, I ate 2,300 calories. I had 200 grams of carbs, 106 grams of protein, 126 grams of fats, and 299 milligrams of cholesterol. A lot of the cofactors that I needed to get the energy to lift 1,104 pounds use vitamin B as a precursor. For example, vitamin B3 or niacin is a precursor of NAD+. Vitamin B2 or riboflavin is a precursor of FADH. Vitamin B5 or pantothenic acid is a precursor of coenzyme A. Vitamin B7 is a precursor to biotin. All of these guys are reoccurring characters, so please remember them. There are also a lot of other important vitamin Bs that produce cofactors like tetrahydrofolate, pyridoxal phosphate, and thiamine pyrophosphate, but if I take the time to go through each pathway that needs them, we will be here all night, so you can just forget about them for now. I wasn't entirely sure how to see how much vitamin B I was getting in my diet, so I looked up things with vitamin B and I got a list. So here are some foods that I ate that might have vitamin B in them, including yogurt, chicken fried steak, broccoli, and cheese. First of all, most of the reactions I'm about to talk about are catalyzed by enzymes. Enzymes usually bind to substrates with high affinity and specificity and increase rates of reaction by decreasing activation needed to make a reaction move forward. Enzymes are not a part of reactants or products of a reaction, they're just helpers. Sometimes they feel left out, but it's for the greater good, so they accept it. Some kinds of enzymes include oxidoreductase, which catalyze, shockingly, redox reactions, transferases, which transfer functional groups, hydrolases, which form 2 products by hydrolyzing a substrate, lyases, which cleave bonds but not through hydrolysis or oxidation, isomerases, which move functional groups around, and ligases, which form bonds using ATP cleavage. Enzymes do all kinds of things to mediate reactions, including facilitating metabolic transformation reactions, as mentioned previously, allowing for redox reactions, and creating reversible covalent modifications. Stay tuned to the end of this video to see which enzyme won the Objectively Best Enzyme Award, which is a highly prestigious honor in which I name one enzyme the best enzyme based on which one I like the most, and then explain how it works so that I can get credit for this assignment. To gain the energy I needed to lift 1,104 pounds, I converted a lot of different food sources into ATP, which could be used for energy. We'll start with carbohydrates. I ate a total of 206 grams of carbs. There are 3 groups of carbohydrates, including simple sugars, polysaccharides, and glycoconjugates. I'll mostly be focusing on polysaccharides and simple sugars. In plants, carbs are stored as starch, and in animal, carbs are stored as glycogen. Most of the carbs I ate were consumed in starch form, degraded by enzymes like amylase to simple sugars, and then digested in my small intestine. From there, carbs made their way through glycolysis, which includes lots of steps that I learned last semester and still totally remember, but the main gist of the thing is that each molecule of glucose is converted into 2 molecules of pyruvate. After that, the pyruvates are converted into acetyl-CoA with the help of CoA, and then they enter into the citric acid cycle. The purpose of the citric acid cycle is to create a bunch of cofactors, including our good friends NADH and FADH2, by moving electrons around through redox reactions. Each turn of the citric acid cycle also results in 1 GTP, which, as far as I'm concerned, is basically the same thing as ATP. After that, the electron-carrying cofactors went to the electron transport chain. The electrons from the cofactors were transferred through 4 complexes on the inner mitochondrial membrane, and as they moved, hydrogen ions were either moved through the inner membrane into the inner membrane space or used up in the mitochondrial matrix. This created a concentration gradient, where there's a higher concentration of protons in the inner membrane than there is in the mitochondrial matrix. The protons on the high concentration side of the inner membrane move through ATP synthase to get to the low concentration side and establish equilibrium. Their movement creates the energy necessary for ATP synthase to create ATP from ADP in a phosphate group. This is where the majority of my lifting power came from. Any excess carbohydrates I ate during the day were stored as glycogen. To do this, I added glucose 1P to the non-reducing end of a glycogen molecule with the help from the enzyme glycogen synthase. The hormones insulin and glucagon, who are my favorite enemies to lovers couple, work together or maybe fight each other to synthesize and degrade glycogen as needed. Next, let's talk about fats. To break down the 126 grams of fat I ate, my body broke the fats down into fatty acids and glycerol. The fatty acids were then transferred into the mitochondrial matrix via the carnitine transport cycle, catalyzed by carnitine acyltransferase 1, and reattached to acyl-CoA for further degradation. Fatty acid degradation consists of 4 repeating steps: an oxidation catalyzed by acyl-CoA dehydrogenase, a hydration catalyzed by enoyl-CoA, another oxidation catalyzed by 3-hydroxyacyl-CoA dehydrogenase, and a thiolysis catalyzed by beta-ketoacyl-CoA thiolase. Each cleavage every 2 carbons results in 1 NADH and 1 FADH2, which are used in the electron transport chain. The leftover acetyl-CoA is put into the citric acid cycle, where it continues to be turned into ATP. Enzymes are used to isomerize unsaturated fatty acids so that they can be cleaved normally. Odd-numbered fatty acids result in leftover propanoyl-CoA, which is converted into succinyl-CoA. Succinyl-CoA is an intermediate in the citric acid cycle, so it can be used to form even more ATP. To make fatty acids with excess acetyl-CoA, another 4 repeating reactions occur. But first, the acetyl-CoA needs to be converted into malonyl-CoA. This reaction is catalyzed by acetyl-CoA carboxylase, which is the rate-limiting step of fatty acid synthesis. This reaction requires our good pal biotin. Fatty acids are then produced with fatty acid synthase, with a condensation catalyzed by beta-ketoacyl ACP synthase, a reduction catalyzed by beta-ketoacyl ACP reductase, a dehydration catalyzed by beta-hydroxyacyl ACP dehydratase, and a reduction catalyzed by enoyl ACP reductase. The cholesterol I ate is used to maintain fluidity homeostasis of my various phospholipid bilayers, as well as for synthesis of cell signaling molecules. If the cholesterol I ate wasn't enough, my body would make more in a deeply complicated way that boils down to 4 stages. These stages include formation of mevalonate from acetyl-CoA, formation of isopentyl diphosphate, formation of squalene, and cyclization of that squalene. Finally, let's talk about proteins. Dietary proteins, like the 106 grams I ate, are degraded into amino acid by lysosomes or by ubiquitin tagging. These amino acids are broken down further into pyruvate, acetyl-CoA, alpha-ketoglutarate, succinyl-CoA, fumarate, and oxaloacetate, which, guess what, feed back into the citric acid cycle. Unfortunately, something needs to be done with all of the leftover nitrogen from the deaminated amino acids, so it all gets peed out through the urea cycle. Because I'm always well hydrated, this is fine, and I agree to this. One very exciting little crossover here is arginosuccinate, which is an intermediate for the urea cycle. Arginosuccinate can be converted into fumarate and then into malate, which can feed back into the citric acid cycle again, depending on cell needs. Most importantly here, we have amino acid synthesis, aka how I got so swole in one day. To assimilate nitrogen, glutamate and glutamine are used. Glutamine synthetase uses ATP and ammonium to produce glutamine. The nitrogen and glutamine, in conjunction with alpha-ketoglutarate in a ping-pong mechanism, provides the amino group for the rest of the amino acids, which are derived from metabolic intermediates. Humans can't make some amino acids, so we have to eat them. These are called essential amino acids. The protein I eat is broken down into amino acids, and then the amino acids are built into new proteins that make up my massive muscles. And now, for the moment you've been waiting for, the Objectively Best Enzyme Award. After long and difficult deliberation, I've decided that the best enzyme is chymotrypsin! Chymotrypsin uses a catalytic triad of amino acid residues, including histidine, aspartate, and serine, to form a network needed for catalysis. This enzyme uses the amino acids to participate in covalent catalysis, as well as acid-base catalysis, stabilizing and altering the substrate. Congratulations, chymotrypsin! Thank you so much for joining me today, and I'll see you next time when I do something else easy like curing cancer or solving climate change.